Yeah, last but not the least, for ending this theme on AI and biology, we have Jen Listgarten. Professor Jen Listgarten is at UC Berkeley in the Department of Electrical Engineering and Computer Science. I've known Jen for a number of years since we overlapped at Microsoft Research New England. She's also in the steering committee of the Berkeley AI Research, the Chan Zuckerberg Investigator, and she was at Microsoft Research from 2007 to 2017, and PhD from Toronto. And yeah, she has a very interesting interdisciplinary background with undergrad in physics and computer science, and also doing now biology. So yeah, take it off with Jen.
I was told there's a pointer, but I guess there's no pointer. How do I work the pointer? It is the M. That is not a. Whoa. All right, there is a pointer. Great. Okay, it's a real pleasure to be here. This is quite a marathon session, so I hope we can all stay awake for this one. I'll try my best. Right, I'll just gonna give sort of an overview of where protein engineering is with respect to machine learning in the beginning of the talk. Then I'm gonna give you just a flavor for some of the projects that my group has focused on in protein engineering and machine learning. I'm a machine learning person. I don't have a wet lab, but I collaborate with wet lab people. And then I'm gonna go into slightly more detail in the last part of the talk into one project that I'm very excited about. So, okay, so you probably know that proteins are everywhere. So not only are they the sort of major workhorse of what goes on in the body, but they touch on things spanning therapeutics, the environment, and so forth. I've highlighted some examples here. I think we all know about antibody-based therapies. You've probably heard about gene therapy, and I'll talk a little bit. That's going to be my case example at the end. We know about CRISPR-Cas9. So that big Pac-Man up there that actually pulls apart the DNA. This is a protein, and people are actively trying to engineer it. A new project I've just started on is dealing with this PET ACE protein. Oh, this is a weird pointer. Okay. Which can actually, like all those plastic water bottles, which are remarkably not in this room. If one could engineer this better, that would be great. And then also this Rubisco is actually the most abundant protein on the whole planet. And if we could engineer it in certain ways, it would be very helpful for carbon dioxide sequestration, for example. So that's just to give you a sense of the broad scope of problems for which protein engineering can have impact. And so why, at a very sort of a simple level, why is this a really hard problem? So we have 20 amino acids in the alphabet. And so one of these, sorry, really disorienting pointer. I'm going to just use my hand. Okay, so right, each line here is a string of made up of the 20 amino acids in the alphabet, and most proteins are a few hundred, let's say, of these. But you can see from here that already at 100, we far exceeded the number. If the length of the protein were 100, how many possibilities are there? This already vastly exceeds the number of atoms in the universe. So at a fundamental level, this is like a nearly infinite search base. So how can we hope to navigate this? So where have there been some successes in navigating this space, and how have they worked? So first and foremost, we have natural evolution, which has spanned millions of years and very, very slowly been able to create proteins that are able to start in this sort of string, in a sense, and then fold themselves. You've probably heard of the structure prediction problem, which is to go from a sequence to folding into a 3D structure, which determines the function that it has. And so nature has done this. And of course, we'd like to build things that nature didn't create, which is why we have protein engineering. And there's a number of protein engineering strategies. And so let me take you through them. So up until quite recently, there were two main strategies. One that was primarily computation-based. I say data-free in quotes because there's a little bit of data that feeds into this, but nothing like the modern sense in which we think about using data in machine learning. And so this is based on a fairly simple but very, very powerful biophysics-based energy function. And that's sort of the key technology, is how do we use this energy function to design proteins? And there have been many successes with this. It's pioneered by David Baker, and it says almost rest in peace. So David Baker did himself take quite a while to get machine learning into his group, but he has. And so he himself, all these papers that he currently publishes, mostly don't use this energy function, but there are some corner cases where people are still using these energy functions. And the second quite complementary way that people have done protein engineering is in the wet lab with directed evolution, which got the Nobel Prize, of course, in 2018. And Francis Arnold, who is at Caltech, was one of those recipients. And here you basically repurpose evolution, right? You don't just let it evolve according to the fitness that natural evolution, you impose your own fitness in an iterative manner in the wet lab. So you start with one protein, you induce a large number of variants through a stochastic process that changes this one starting parent protein. And then you measure for the thing that you care about in the laboratory, and you take off, say, the top 10 performers according to this thing. So you have to be able to measure it. And then you kind of go through as a computer scientist, you could think of it as a greedy stochastic search through there, and you get to a local optimum. And if you're lucky, you may find the protein that does what you want. And then, of course, why are we all here? Because machine learning is infiltrating all of the sciences. And so in the past number of years, machine learning has come into play in a number of ways throughout everything I've just said. And so that will be the topic of the rest of the talk. So sometimes I get this question. They say, people kind of a bit outside of this area, they say, well, AlphaFold is there and it kind of crushed the protein structure prediction. So like, why are you still working on protein engineering? And so I'd like to address this question, which is, did AlphaFold 2 solve protein engineering? And so unambiguously, no, it did not. Unambiguously, it's transformative. It is a breakthrough. It was really a stunning success. And so if you've never seen this plot, this is sort of the cast competition over the years. And the last one is when it really had a phased transition, if you want. In this case, actually, the previous AlphaFold wasn't beating the other models, and it wasn't as big of a jump, although there is still a decent jump. So why not? So what does AlphaFold do? You give it a sequence, and it tells you the structure. So that's great, and it can be super helpful when we're trying to engineer proteins. But fundamentally, we don't know what sequence we want, and we don't know what structure we want for a particular function. So suppose you have a target and you want to design an antibody to bind to it, you can't really actually use that, right? So if anything, maybe you have an idea of the structure. Actually, often you don't, but just suppose you did, then you'd have to use the inverse model of AlphaFold. You'd give it the structure and say, you tell me the sequence I can make in the lab. And this is now sometimes called inverse folding. And in fact, one of my students at an internship did reverse. They ran AlphaFold and learned the opposite model to invert it. And this turns out to be also quite useful. But in my opinion, the bottleneck challenge here is that we don't know how to predict the function we want, either from sequence or from function. And to the extent that there are successes in protein engineering, usually they're grabbing out a site from a protein that already does what they want. And then they're just saying, I want to make it smaller, or I want to do this, or I want to do that. In terms of the biophysics-based approaches, directed evolution is able to kind of do more novel things in this totally different way. So hopefully you now believe AlphaFold 2 is pretty amazing, but not solving the problem. Okay, so what are the problems that people are thinking of in computer science or in the wet lab? So if you just hear protein engineering and you read this paper and they say we've solved this problem, and it's very disorienting. There's a number of problems at play here. So I just want to kind of paint a picture. I wish someone had actually painted this picture for me when I started in the field only about 5 years ago. So fundamentally, we have sequences. Those sequences fold up into a 3D structure, and that structure induces some function. Which also may have to do with the specific amino acids in that structure. And so you can think of predicting the function from the sequence. We sometimes call this fitness prediction or function prediction. You can think of predicting the structure from the sequence. And so these are prediction tasks, all of which people are working on. And then sometimes these get called as the design task. So suppose you have, so the most common one you'll hear is that someone gives a structure and says, tell me the sequence. So this is structure-based sequence design. And sometimes you do backbone design, which means you're specifying some properties and you want to find the 3D structure that gives you those properties. But even if you do that, you're not done because you still have to find the sequence. So all that is to say that all these different tasks are important. People are working on different parts of them. And often a paper is just about one of these. And even if they've done a great job, doesn't mean that they've solved protein engineering, but there are many wonderful papers, of course. So, okay, so now I just want to go through a few high-level trends that are in protein engineering that are happening right now. So a big one, of course, not only in protein engineering, but is representation learning. And so what people do here basically is they grab a giant database of naturally existing proteins and they put it through an unsupervised, or if you like, self-supervised model. And they basically try to get imagine you push it through a neural network to predict itself, and then you take out one of the layers that's like of fixed length and smaller than the protein. You treat this as a representation in the hopes that it's sort of capturing the manifold of all naturally existing proteins. And you hope that this representation, instead of the raw amino acid sequence, is more useful for machine learning. So this is one type of a trend. Another one is to build generative models. This is a very topical, very timely topic in general. But so, and there's two ways. On this slide, I'm talking about generative models for sequences. And so one thing we want to do is just be able to unconditionally generate things that look like real proteins, that behave the sort of laws of physics that someone who's a protein expert would say, yeah, that looks plausible to me. But we may also oh, sorry, this is sequences. I got my slides mixed up. We just want to generate sequences that would fold up into proteins, and we may condition it on a particular structure. That's the structure-based design of sequences. And some people have started to play with conditioning on a control tag, which is a word used from natural language processing, where you would actually give it an English label, like this comes from the cores made mutase family. Now make me a sequence that belongs to that. I personally think that latter one is a lot less interesting and useful. And you can ask me why at a coffee break. Okay, so another one, and this is actually probably the one that's getting the most attention right now. There's some few papers almost competing with each other on generating structure, the 3D backbone structure of a protein. And they're focusing on how to do this in a very general way, such that it is a protein that it could be a real protein, and it's not kind of just looks nothing like a protein in terms of the angles between various parts of the physical structure. And then even more importantly, we want to do conditional generative design for structure. And so something that gets missed in a lot of these papers is that if you want to do conditional generation, so of the backbone conditioned on, for example, F is some function, suppose that function says like bind to that target there, that you can only do that as well as you can predict the function from the backbone. So if you're familiar with diffusion models, you need generally to guide the model with this kind of a piece of information, because you don't get something for nothing, right? You can't just kind of throw the whole protein universe into an unsupervised model and expect it to know much about specific functions like catalytic efficiency, fluorescence, binding to targets, and so forth. So I think this is a very powerful direction, and there are some particular cases that it currently does work for, but I think there remains to be a lot of work on this particular aspect here, which is what we sometimes call function or fitness prediction. And so here, unlike natural language processing and computer vision, we have very, very little labeled data, right? So you can label computer vision data on anyone in this room, but to get that kind of information for 100 proteins with a biophysical assay is a crazy amount of time and money. And so we're in a very different situation. However, we do have access to some information that they don't have really, I would say, which is evolutionary information. And evolutionary information is driving a lot of the progress in this field. So in particular, it's actually, you may not know, but inside of AlphaFold 2 they're standing on top of many years of work of how to use evolutionary information to fold a protein from sequence into structure. And that same kind of information can be used here to build what we call in machine learning zero-shot models. It means, how can I predict this function at all from the sequence when I have no labeled data? And so I'm not going to go into the details here, but there are ways to sort of squeeze out of the natural databases evolutionary signal that gives you a set of proteins that are probably enriched for certain functions that you care about, that you can then model. Okay, and so the last but not least, on the one hand, everybody says structure prediction is solved. So it kind of is solved and it's kind of not solved. So there are some very important corner cases. So something people are actively trying to tackle is how do you get the structure from a sequence for a protein, which is what we call an orphan protein. So an orphan protein is one that doesn't have evolutionarily related proteins in the database. And because AlphaFold depends so heavily on that information, it does not do well for proteins that sit outside of that space, these orphan proteins. It's also not very good at understanding how two proteins that come together and undergo a conformational change to bind, it's not good at that either. If they don't have to undergo a conformation, it can be pretty good. And then finally, I think Anshul probably alluded to some of this in his talk. It's not very good at protein DNA binding or protein RNA binding. This is important for a whole host of problems, including gene editing and so forth. Okay. So now I'm going to just go over sort of a one-slide view of a few projects in my lab before I dive deeper into the last one. So, okay, so those are the sort of mainstream problems. I don't like to work on the mainstream problems, but I really like this area. When I started to work in it, I didn't think it was a mainstream area, and then it very quickly turned into one. So we've been sort of finding our own corners, let's say. So we've been very focused on this problem of how can we predict the function, how with machine learning and using evolutionary information and anything we can bring to bear. And if we want to now use that function, this prediction of the fitness from the sequence, when can I trust it? How do I know if I can trust it? Because if I'm trying to design a protein to do something I currently don't have any proteins that do, then I'm sort of by definition extrapolating, right? I'm outside of the training zone. And so a lot of the themes of our research have taken into account or are thinking about this natural tension between pushing the model, this fitness model or function model, to parts of the space that have property values we want, and at the same time realizing that we can probably only push so far before that model completely breaks down. So that's a big theme, and I'll mention a few. And another one, so one of my pet peeves about this field is that what are the big giant models for protein engineering called? They're called giant language models because much of what people have done is to literally port over from NLP a bunch of architectures and technology into proteins. It's not exclusively the case. There are some nice examples where it's not the case, but I would say in general, not a lot of thought is given, right? The latest diffusion model comes out, the latest this comes out, and you like guaranteed within a month, there's a preprint that has that for this problem of all those problems in protein engineering. And so I think it's sometimes nice to just go back and say, what if we had designed these things for proteins? And so we have a little bit of work in this space, and we're thinking a little bit more. And I think I took that slide out, but roughly speaking, you could think of, we're trying to think of what's an appropriate basis in which to represent functions in which we want to have sparsity. And so technically, it's a discretized version of the Fourier domain, but I will not go into this. It's like a teaser if that meant anything to you. And then the part I'm actually most excited by that I will focus on at the end is often we think of protein engineering as give me that one sequence that's going to serve the purpose that I want. Like it's going to bind that, that SARS spike or something. But in reality, we need a bunch of lottery tickets. We need to like a diversified set of hypotheses that we're going to go test in the lab. And depending on the protein you're working with, depending what's known about it, depending on how well you can measure the property you care about, there can be a range of scale at which you're going to measure things. But in almost all cases, you're generating hundreds to millions of candidate sequences. And so in our collaborations, we're working with people who actually generate large libraries of proteins, which is in the millions of proteins, and they generate them with stochastic knobs. And I'll talk a little bit more about that. And as machine learning people, we get to control the knobs that generates a distribution of proteins that we hope are good candidates to pass the selection. Okay, so I really like this analogy about what can go wrong if you have one of these function predictors. So something that takes a sequence and predicts the function, like will you bind to the spike protein of SARS-CoV-2 or something like this. So the way we usually think about machine learning, right, is we're giving inputs X and we're paired with outputs Y, and we fiddle with gradient descent to set the knobs of this neural network. But in the problems that we're thinking of, what I now call machine learning based design, we're inverting this problem. So what we're doing is we're saying, suppose someone gave us the function predictor, they've already learned it to the best of their ability, and now I want to design something by telling you the output I want, and you're going to figure out the input, right? This is in a sense what design is, and you can do this in a number of ways. You're essentially inverting the model. And so this, I apologize, I forgot some of the credit. I think this is from a Google blog where what they had done, so I did not actually make up this example, but what they did is they trained a giant neural network to predict classes of objects like banana, peach, apple, zebra, like hundreds of things. And the question they had was not to actually design a banana, so I'm slightly abusing it, but they did actually do a design procedure. What they wanted to do is understand, what does their trained neural network think a banana looks like? And so if at a conceptual level, the machinery is the same. What they do is they take the trained network, they set the banana node to 1, and they set all the other object categories to 0. And then they use backpropagation on starting with a random image and see what happens. And then this is what happens is you get abstract art. You cannot go 3D print this, right? And so, so that tells them something about what's happening in their neural network. But for us, if we specify that we want catalytic efficiency to be high and we reverse this network, we're going to get similar garbage and it's going to be totally useless. And that's because, in a sense, we're squeezing these models for all they're worth and we're pushing them into parts of the space that they were never trained to use. So another analogy here is, you may have heard of adversarial machine learning, where an adversary tries to fool a classifier. So you can go through a pretty simple process to force a very confident neural network to think that this stop sign is actually a yield sign with super high confidence. And you just do a clever thing to add some imperceptible noise to push the neural network into this pathological space where it thinks it knows things and it's just completely wrong. So we don't have an adversary, but in design, we're our own adversary for squeezing into the extrapolative part of the space. So, okay, so now I'm going to go through just some of the ways we've been thinking about this problem. So it's actually a fairly simple technique here, which was in this paper talking about how would we invert a model so that we don't end up with this problem. And in some sense, there's not a magic bullet here, right? You have to kind of have some notion of where you trust the space or not. And I think there's some reasonable ways to think about that. But more importantly, in this paper, like, no one had even been thinking about this. They were treating protein engineering as optimization, and then they were just comparing optimization algorithms, ignoring the decades, centuries of optimization research, and then publishing it at a protein engineering conference. But in any case, so we just brought this problem to the forefront and thought about it a little bit. And so basically we developed a coherent way to invert this model while paying attention to sort of side information in a way that's integrated with the model inversion. And so at an intuitive level, like, where might you get data that would help you avoid this problem? Well, fundamentally, you want something that basically says, is this thing protein-like or not? Like, is it going to just stably fold into something? And so how might you get that? You might get that by taking actually some of these large language models can actually, under the hood, even though they weren't intended to do this, this can be what they pick up. So you can use one of those to estimate a density that you can use as a soft trust region. And then fundamentally, you probably want to pay attention to where your training data lay as well, right? So whatever sources of information you can think to layer in here are probably going to help you. In practice, what people often do is they just say, don't go more than 10 mutations away from a protein I know, or something like this. Okay, and then right. So Hunter had this insight that even if you're using machine learning, that you may want to still use something like a biophysics-based model if one exists and it's reasonably good. And so, well, why would you do that when we've just said that, machine learning is kind of taking over these biophysics-based approaches? But if you think about it, machine learning, so this is like a simplified example. So these red data points are like training data, and we fit the function there. And so probably by having data there, a machine learning model is going to be better at predicting that function than some biophysics-based model because we got training data. However, as I get further away from the training data, it's almost certainly the case that these biophysics-based models will do better. And so now there's this question of how do I blend these in a way that lets me sort of optimally explore extrapolation space? And so he came up with a method that's meant to, again, just be sort of a rigorous, coherent way to blend these things based on certain kinds of uncertainty. And and this is a different one. So this is Clara, who did this all while having a child in grad school, which is just amazing. And so there's a kind of a trendy area in some pockets of something called conformal prediction. And really what it is it's a way to get guaranteed confidence intervals. But she really is interested in protein design, and she liked the sort of underlying technology of conformal prediction. So for example, if this is my sequence, like give me the exact guaranteed confidence interval that it has this particular, say, scalar property of binding to that thing. And so, but she wanted to be able to do it in an iterative design cycle where the test data actually depend on the training data, because when you do design in an iterative manner, you build a predictive model and then you use that model to design the next round. So you have this dependence between the test and the training data. And so she generalized this sort of formal area in order to have it be applicable to iterative experimental design. And I think the nicest thing that came out of this is it's not particularly useful to be truthful about particular predictions, because although it is guaranteed and it's finite sample and with no assumptions, there is one assumption, which is an expectation over all data sets, which is kind of a little bit funny. But in any case, what is on this picture here is meant to highlight what I think one of the main strengths of this work is, which is to be able to set certain parameters you don't know how to set when you're doing design. So what do I mean by that? I said that when we do design, we don't usually just generate one protein sequence. We typically generate a whole library of sequences. And we think of these sort of as lottery tickets, and we want to diversify our hypotheses, right? So we want a diverse set of good candidates. So usually in our design process, there's a knob that controls the diversity of the sequences. So we want them to have high probability to bind that spike protein, and we want them to be diverse, because otherwise they're all going to look almost the same, and there's no utility in doing that. We are wasting our experimental budget. So what this shows is that with these confidence intervals, you can basically see how as you make your library have more and more diversity, you're going to go in, so that's increasing lambda. As you do that, so actually, which way? Mean predicted. Suddenly I got confused by this plot. As you have less diversity, you get higher predicted. You know what? I'm suddenly confused. Let me skip this. Wow, that's super weird. Oh yeah, sorry. Okay. So as you go further into extrapolative space, then you get higher fitness and you get increasingly wider confidence intervals. So you're less certain because you're going into extrapolative space. And so basically using this as a tool, you can start to play around with how might I change the diversity while making sure I'm not going into kind of crazy pathological territory. So it's not telling you what decision to make, but it's giving you a tool to make that decision. And then this was an accidental paper that I think just had a couple of good takeaway points. So what happened is a bunch of papers started coming out doing taking large databases of proteins, pushing them through the giant models. In this case, the one that motivated us was an LSTM. Now, of course, it would be a transformer. And then they were using this representation to do a very lightweight regression, like a linear regression on top of that very nonlinear representation. And they were making claims that this was very helpful for protein engineering. And it was creative and it was new. And it was not at all clear if it was really buying you something, because what happens in a lot of these papers is by the time it gets published in Nature or wherever, they have produced a protein. And so there's like an experimental validation, but you don't actually know what other simpler computational methods might have led you there. So that's not always the case, but it is often the case. And it makes it very hard as a computer scientist to understand whether they have done something truly useful or not. And so this project, we were just trying to dig into these things. And so Chloe also got excited. And so she just started to run a whole bunch of comparisons on certain tasks that have to do with function prediction, either just from evolutionary data or when you also have some labeled data, and say, like, did these complicated models really do something? And so actually, in this particular case, at the end of the day, those complicated models were beat out by very, very simple models. And so part of the story here is just like people, please just do some simple baselines so that we know what's useful or not. And then the other thing is, actually, I always tell people, even our paper is garbage. Why is our paper garbage? It's still the best you could do at doing this in-depth exploration, which many people appreciated. The reason it's garbage is that all of these plots are based almost entirely on a very strange test set. They're based on test set where the proteins that are being tested in the predictive accuracy are just a single mutation away from the sort of central wild type protein, the naturally existing one. And so if you think that, very complicated machine learning is going to help, but your test set is like one mutation away from what you know, like you can't even really explore this. And so in some sense, it's not surprising that we got the results we did. Of course, this is all changing right now, even though this is 2022, this is completely out of date right now, but I think it's still a good lesson for the community. Okay, so that gives you a flavor of some of the things that we've been doing. And so we also have a number of ongoing wet lab collaborations. And I want to, in closing, just talk about the last, or the first one here, which is the longest running one we've had, which is centered on a gene therapy example with David Schaefer. I brought water. Okay, so Dave and his lab are experts in directed evolution and in bringing proteins to the clinic for therapeutic purposes. And he's a, oh, thank you so much. And so this has been a several-year project. And so the point here is that there's this non-pathogenic virus, adeno-associated virus, that almost everybody in this room has been infected with. And that's because it's innocuous, so it doesn't really matter. But viruses are very good at getting into cells, right? So they're a very good, promising candidate as a delivery mechanism for gene therapy. And so that's exactly, many people are trying to use AAV to engineer it to do specific types of gene therapy, meaning to put it into particular cells in the brain or elsewhere. And so, in fact, there are many very promising clinical trials. In fact, Dave is sort of independently wealthy now as a result of a company in this area. However, there are many disease targets that we still can't hit. The ones that currently work are very corner cases. So for example, they're working on cells in the brain for which it's difficult the immune system to get there. And so one of the problems is that because we've all been exposed to these, the immune system actually just goes and shuts it down before it can do what you want. So something we want is to figure out how to change them so that they don't get hit immediately by the immune system and still are not toxic, for example, right? So, but there's many other issues that also need to be overcome to have this be a more general purpose gene delivery mechanism. So it's very, right now, there's certain actually cells in the brain that they literally just cannot get them to hit, which would be very important for a number of therapeutic and basic scientific questions. Or it's not specific, so maybe it does go to the cell type, but it also goes to 10 other cell types you don't want your drug delivery system to go to. And so a number of things like this. And so when I first met Dave, what we decided to do is to start with a very simple project, and we're now layering on top of it. But I really, even though it's a very simple project, I just really like it. And so the other ones are not as mature, and I'll tell you about this one. So the idea was that the way this works is he's a directed evolution guy. So they start with a giant starting library that generates millions of peptides. So they're not generating the whole, this is actually several copies of the protein that are all together to function as a unit. And what he actually, through the decades of work with this, what he and other people know is there's a particular region on the outside of the protein, the capsid, where if you insert just 21 nucleotide insertion, it has dramatic effect on all the phenotypic properties of the virus. So by changing this 21 nucleotide insertion, you can change what cell type it goes into, you can change its immunogenicity, many, many things. And so it's a good diversity mechanism to create many, many versions and check which ones work. And so this is how they do things is they start with a library that's uniform over those 21 nucleotides, and then they go through rounds of selection to try to find what they want. And so the question was, what are the different ways we could bring machine learning to bear here? And the very first one was that it turns out that the starting library, half of it is wasted. And so actually the first round of selection has to fix this problem. What is the problem? It's just generating capsids, proteins that don't work. The whole thing just kind of falls flat and you can't use it for anything. So you're just, from the get-go, you're wasting half of your budget. And so part of that is, sometimes people say, well, just do that round of selection and then you fix the problem. But you haven't, because after that round, you've lost so much diversity that you've lost so many lottery tickets that for whatever your downstream problem, you probably won't be able to tackle it nearly as well. So what we said is like, let's just design a new general purpose starting library so that for whatever property you want to engineer it for, even if we don't yet know about, it's going to be a better library. So I have mentioned this word library and I've alluded to this idea, but just to make it clear, because it's important for this project. So we think of a library as a large set of proteins that gets generated in the lab. But for us as machine learning people, it's a probability distribution for which we get to control the knobs. And in this very simple example here, this is 7 amino acids, so 21 nucleotides. I wish I had the other version of the plot, and basically we get to control the marginal probability at each position for any of the, it's actually at a nucleotide level, but we just get to say at every position, I'll tell you the distribution of ACTG. And by controlling that, we generate a different, as we change it, we have a different distribution of 21 nucleotide insertions. And so in the beginning, that's flat as the current starting library, and we want to make it not uniform so that it has the highest possible amount of functional proteins while being as diverse as possible. So that's basically our goal. And so we've done this. This is it's a few slides, but it took a number of years. So we took the starting library that everybody in the world uses for this problem, this uniform library, and then we measured how well, how viable, technically, how well it packaged each version of the virus through next generation sequencing. So now we have essentially supervised data. We have for each 21 mer insertion, we have a label of how well it packaged. It's actually approximate because these sequencing based ways of reading things have some problems. But it's the only way you can do things at scale. And so now we build a simple supervised model. There's nothing interesting in this particular machine learning model. It's just a small neural network. And we would try a number of them and we pick the one that works best in a cross-validation. And then, as I just said, this way of measuring things with these sequencing reads is not quite accurate. What we really want are to measure what is called the viral titers. It's like literally in a very low throughput manner, how much of the virus is there. So this is the real thing we care about. So we wanted to make sure that our model could predict that. And so we picked a spectrum of sequences and put them through that were not in the training data, put them through our model to make sure that actually when we measure the real thing, this is what that it makes sense. And indeed it does, we get a very beautiful correlation. So then the next thing we did, and this is an overlooked part of protein engineering, I would say, is how to think about how to trade off the diversity versus the property you care about. So what I mean is that I'm going to pick the knobs of this distribution in a way that I want all the viruses to package. And so actually at the topmost up here, these are the ones that all of them are going to package, but they're going to be almost all the same because there's no diversity. So this is a waste, right? Like if I have 1 or 10 proteins when my budget's actually 1 million, then I'm not making good use of it. This is the standard state of the library point here, which has great diversity and has terrible, terrible expected fitness. And so what we can do is we can trace out a Pareto optimal curve and say, well, let's get somewhere else on that curve that's more useful. And in general, these curves, you don't necessarily know where you want to be on a trade-off curve. In this particular setting, because this is vertical, this is unambiguously better, right? It has the same diversity and vastly better fitness. And so we actually made a couple of these libraries. And in the end, we actually picked the D2 library is the one that Dave now uses in his lab for all his experiments. And so the first thing we did was say, well, we have an expected average fitness for each of these libraries. Again, let's go measure in this very detailed manner in the lab if that's actually what bears out, and indeed it is. So basically, we've now validated that this curve is giving libraries that have the properties we want. And then finally, this is really the big punchline here. So the punchline here is now we said, let's prove that having designed this new starting library is useful for a downstream function, a downstream selection that it was unaware of, right? We're basically just trying to get really good lottery tickets for whatever the competition is. And the competition here was to infect brain cells, 3 different types in the brain. And so what we did is we tried the standard baseline library, which is basically uniform, and asked how many are able to infect these cells in the brain. And then we put in our machine learning designed one. And I won't go into the details of exactly what these numbers are, but more or less this is like, it's about 10 times better, roughly speaking, at finding AAVs that can infect those cells. And so this kind of idea can be generalized beyond this in many ways. This is just the first very simple example. So it's just 21 nucleotides. We have control only over the marginal, but you can get to much more interesting probability distributions with much more complex knobs that touch on much larger parts of the space. And this is some of our ongoing work. So I'm going to wrap up everything here with some parting thoughts. So I think these are I've actually meandered in my career through all kinds of problems. I've been working in proteins always with machine learning, but I've only been in this about 5 years. And I really am most passionate about this particular application domain compared to all the other ones. So I think it's very exciting. And of course, people are now, the buzz is I'm sure there's at least 5 startups now that are probably super well funded where their pitch is probably ChatGPT-4 for protein engineering. And I'm personally pretty skeptical about that being around the corner. So why? I mean, we just have vastly, what are they doing? They're absorbing the world as it appears online and in textbooks, right? But you can't, this information doesn't exist there. So it's just not going to happen. And we're going to have to be much more clever to make big leaps in this field, I think, than in some of these others. But I welcome pushback on that. I'm very curious to hear. And I think AlphaFold 2, although it does not solve protein engineering, it is unambiguously a great win for the field on the whole. And I think this idea of predicting function is often a bit overlooked in this overall area. And even though I am focused on this area, I get whiplash because it's just like every couple of weeks there's a new preprint doing something and you cannot tell what's working. You can't tell what is working. It's just like completely crazy. Okay, thanks for your time. I guess everybody wants coffee, but if you have a question.
Hey, John, great talk. Thanks. I was just curious for the AAV design component. You talked about cell type specificity. It could be very interesting.
About what?
For the AAV design. So you talked about cell type specificity to infect very specific cell types. I don't know enough about this, but you could imagine engineering the virus, but also engineering the promoter of the gene that's being expressed. And if you couple the two, you might actually get incredible specificity.
There's some companies, I think, going in that direction. And I think that there is work in that direction as well. Yeah, that's correct. Okay, I think everybody, there's a question. All right.
You mentioned a couple of different ways of capturing uncertainty, including Bayesian neural networks and conformal prediction. And I just wondered from your perspective, what some of the trade-offs are, or what you think about the two.
Yeah, it's a great question. Partly I feel like it'd be better over coffee, but I think they're all kind of flawed and they're all kind of useful, and I don't adhere to any one of them, I guess. At the end of the day, look at your problem, look at what's computable, see what the assumptions are for different ones. I mean, I think there's a big misperception that so one area of experimental design is Bayesian optimization. And everyone says, well, I'm Bayesian, so my uncertainty is correct. And I just pipe it through an acquisition function. I just solve an impossible optimization problem. So I'm good to go. Right. And so I think they're all more or less like that in different ways. But that doesn't mean that they're not useful in guiding us.
No, that makes a ton of sense, but maybe just to flesh out a little bit,
Yeah.
like in this particular field, if you have to design a prior, how easily do you feel like you can do that?
I think it's really hard to make a general statement for that. And so actually the conformal prediction doesn't require a prior, for example. The Bayesian neural network, so actually the really interesting technical bit of that paper is that Bayesian neural networks implicitly, in a sense, have a prior over the weights. And that's not in any way meaningful for us. And so the challenge here was to say, how can we make use of Bayesian neural networks, which have uncertainty in some meaningful, hopefully, way, when we have prior information on the function value, which is a totally different currency than the weights, right? And so that the problem there was that the currencies didn't match, and we had to figure out how to do that. But you're asking a question of maybe in that scenario, how do you go about getting a prior? And this is very domain specific. It just really depends. There's one chemistry example and two protein examples. And my student just, he just knew which ones to use from digging around. So, all priors, I think the more information they're provided and the more specialized they are. But in the beginning of the talk, I did mention using sort of a prior as a trust region in the, machine learning based design. And I think in those scenarios, you could have much broader priors that are not very specific. They're just is this thing protein like, right? And so it really depends which part of the problem you're trying to inject that knowledge and what problem you're trying to overcome by putting it there.
Beautiful talk, Jennifer. So I particularly liked your paper. I've said this to you in person before about sort of comparing all these different models and sort of a sort of common benchmarking. So I think in Rick Stevens' talk this morning, he showed a beautiful example of these large language models undergoing.
Unfortunately, I missed. So if there's,
I'll recap the point that I was going to make. Undergoing sort of a phase transition with model size. Chat GPT 3 to 3.5 to 4, and showing sort of orders of magnitude improvements. Do you have hope that we will see something similar for protein engineering?
Actually, I think it's precisely the point is that I don't think scale will fix this, right? Scale, they have infinite data there.
That was going to be my question.
Right?
We're just hunting for the data you think is the answer.
We do have a lot of unlabeled data, but that's very different than the sort of highly informative information that are being used by these models. So it could be that scale helps, but I don't think that's going to be a game changer. I think something at a more fundamental level has to be there first. And to be fair, I'm not sure that it will be there ever, but maybe, in a year, since someone will prove me wrong.
Thank you.
So I'm getting out of time. Oh, out of time. Okay, we got to stop. You can grab me after. Okay. I don't know. But nope. I was told nope. They're going to cut my head off. Okay.